Today I'm going to talk to you about nucleic acids. There are two types of nucleic acid that you need to know. The first is deoxyribonucleic acid, also known as DNA. The second is ribonucleic acid, also known as RNA, which we'll come on to later. DNA was first discovered by two scientists named Watson and Crick in 1953. They were later awarded the Nobel Prize for their discovery in 1962. DNA as a molecule is highly conserved, which means it is found in many different organisms. The first are eukaryotes, which are organisms with a true nucleus, such as animals, plants, and fungi. However, it's also found in prokaryotes, which are organisms with no nucleus, such as bacteria. We will discuss these two types of organisms later on in topic 2 when we talk about cells. DNA has unique properties that make it key to all life on Earth. There are 4 main features of the molecule. The first is that it is a huge molecule, meaning that it can store vast amounts of information in a relatively small volume, i.e. the nucleus. The second is it has small variations in its structure. These variations can act as the simple genetic code. However, these variations also mean that the DNA is different in different organisms. The third feature of DNA is that it's stable. This means that the molecule is not very easily corrupted, which makes it very good for replicating itself. The fourth is that it can actually reproduce itself. Therefore, when cells copy themselves, they will gain an exact copy of the DNA from the original cell. Nucleic acids are composed of monomers or single units called nucleotides. Nucleotides are made up of 3 subunits. The first subunit is the inorganic phosphate group, which is represented by a circle. This is joined onto a 5-sided sugar called a pentose sugar. In DNA, we call this sugar deoxyribose. In RNA, we call this sugar ribose. Attached to the sugar is a nitrogenous base. And there are 4 main bases that you need to know. In DNA, there are 4 types of nitrogenous base. The first is the base A, which is called adenine. The second is T, which is called thymine. The third is C, which we call cytosine. The fourth is G, which we call guanine. The bases bond to each other in a very specific way. Adenine always bonds to thymine, and cytosine always bonds to guanine. The bases bond by hydrogen bonds, which form between the 2 bases. DNA is an example of a polymer called a polynucleotide. The reason it's a polymer is because it's made of the same repeating unit, which are the nucleotides themselves. So here you can see that each nucleotide joins together to make a long chain. They are joined by a type of bond which form between the pentose sugar in one nucleotide and the phosphate group of another. We call these phosphodiester bonds, and these are special types of covalent bonds that form. They are formed by a condensation reaction, which we know from Unit 1, Topic 1, which is the elimination of a molecule of water. When the nucleotides join together to form the polynucleotide, this forms the backbone for DNA, which we call the sugar phosphate backbone. Here you can see a molecule of DNA. You can see that there are 2 strands of the molecule itself. This is double-stranded. This is the DNA double-stranded molecule. You can see here that these are the nucleotides, which are joined together by these phosphodiester bonds. And you can also see here, this is an individual nucleotide, which we'll put a box around here. You can also see that there are hydrogen bonds that are formed between the bases. So here is guanine, and here is cytosine, and here is adenine, and here is thymine. Another key feature you'll probably be able to see is that DNA runs in a specific way. Now, you can see here that the top bit here we say is the 5' end. The reason why I call it the 5' end is because the topmost part of the DNA is on the 5th carbon. So if we use our rule, like we do with glucose, 1, 2, 3, 4, 5, that's the 5' end. And it runs down to the 3' end, so we've got 1, 2, and 3. DNA on the other side of the molecule is from the 3' end to the 5' end. So we've got 1, 2, 3, and the other way around is carbon 1, 2, 3, and 5. This is what we say gives DNA its anti-parallel nature, which means the strands run in opposite directions, which is quite important when DNA replicates itself, which you'll learn about in the next lesson. Now we're going to talk about RNA. RNA is very similar to DNA, however it does have a few key differences that you need to be aware of. These key differences make it's important in its role in the role of protein synthesis, which you will learn about at the end of the year. As a nucleotide, RNA is composed of a sugar. However, unlike DNA, this sugar is no longer a deoxyribose, however it's a ribose sugar. It still has the same inorganic phosphate, and it still has bases attached to the sugar. However, there are 4 bases in RNA, but one of them is very different to that in DNA. In RNA, you do not find the base thymine. Instead, thymine is replaced by the base uracil. So how that will look is in DNA, A binds to T. However, in RNA, A will bind to U, or uracil. So adenine binds to uracil. Another key difference between DNA and RNA is rather than being a double-stranded molecule like DNA, RNA is a single-stranded molecule, which makes it very important for transmitting genetic information out of the nucleus. Another key difference is it's much shorter as a molecule than DNA. This is also a very important feature for it to leave the nucleus as well. Finally, I just want to look at a comparison between DNA and RNA. The first comparison is they both contain an inorganic phosphate group. That's a similarity between the two molecules. The second is they both contain a pentose sugar. However, DNA contains the sugar deoxyribose sugar, whereas RNA contains the pentose sugar ribose. The next comparison I want to make is about the base pairs. Remember what we said? Adenine binds to thymine, cytosine binds to guanine. However, in RNA, adenine binds to uracil, and cytosine will bind to guanine. That's all for today's lesson, and I will see you in class.